Okay, people. So the recording has started. So I'll start sharing my screen now. Okay, people, can you see this screen?
Yes, sir.
Great. So this is where we left off yesterday, and the question we asked was that these various studies, they have used different kinds of methods and control methods. They have all used different kinds of models, different kinds of stability concepts, different ranges of parameters, etc., etc. So if one is trying to use something, then which model do they which method do they choose? Until and unless you compare the methods against each other, you can't really have a good answer to that question. And one way of doing such a comparison is what I had underlined over here. So I'll not go through this at all. I'm going to skip this entire part. Okay. And essentially, this was a lot of work. This was about 2.5 years worth of experiments, hell lot of simulations, all kinds of stuffs happening. 3 papers came out of it. uh, the two things, constancy and persistence, we were considering earlier. Two extra things that we brought in were how much of effort is required. So basically, how many individuals you need to add or how many individuals you need to kill, and also what is known as effective population size, which is a measure of the evolutionarily relevant population size in the controlled populations. This is very important because if not if in populations that become small, this effective population size becoming getting reduced leads to all kinds of inbreeding like effects, which causes all kinds of genetic problems for the population. So you really want this to be pretty high, as high as practicable. So anyway, uh, we did all that stuff, and essentially what we ended up seeing was that if you control a population from one side, that is, if you either don't let the population fall below a level or don't let the population go above a certain level, so all these one-sided methods, they cannot simultaneously attain all kinds of stability. However, if you have two parameter control methods, then that is the only way by which you can attain all kinds of stability parameters. So basically what we ended up showing was that two parameter control methods, where you are controlling from both the top and the bottom, those are the ones which are the best of the lot. And then of course we did all kinds of simulations and ended up showing that our results are pretty generalized. But anyway, what we want to really talk about is the stuff with which we started the whole discussion, which was that if you are talking about building mechanistic models, then how does one go about doing them? Okay, so if you remember, we were doing all this in the context of models like Ricker or Logistic or Hassel, which takes one quantity, the realized population growth rate, and then essentially casts everything in terms of that. But as I have been discussing again and again, realized population growth rate is something that, experimentally speaking, you don't have an handle on. You only estimate it by fitting some kind of a model after your entire experiment is over. If you look at it biologically, what matters for in terms of the organism is things like how many eggs it lays, how long those eggs survive, how many larvae are or how many juveniles become adults, how many of them mate, and so on and so forth. So that's what I mean by saying the life history of the controlled populations. And the problem is that you can't really use a one-dimensional model like Ricker to answer these kind of questions. So in order to answer these kind of questions, what you need is, or one of the solutions is, what is known as an individual based model. So in individual based models, what you do is you explicitly track each individual, and each individual can have a set of parameters which can change based on whatever rules you frame, and then you figure out what is happening to the entire ensemble. You figure out what is happening to the entire population, how its size is changing, and so on. So if you have, let's say, a stage structured population, so for example, you know that Drosophila is stage structured, right? You have eggs, you have larvae, you have pupae, you have adults. Then studying the dynamics of this kind of a stage structured population is obviously not going to happen if you have a Ricker kind of a model, simply because it doesn't incorporate stages. So for that, those kind of models, one of the solutions is what we had, that a Leslie matrix kind of a thing. This is the other way of going about handling the same problem. Now, individual based models, unfortunately, because they are incorporating a lot of realities of the biology, therefore they no longer remain generic like the Ricker. Okay, they become tied to a system. So what we are going to talk about is an individual based model for Drosophila dynamics, and in the context I am going to also show you how the biology can be explicitly brought into picture. So in this, we are going to look at how the life history traits, they interact with availability of nutrition, and why nutrition, will come to that in a moment, and then how these two together shape the dynamics of the population and its stability. So in what way the numbers change over time, and how to reduce their fluctuation, and so on. So as you know, the life cycle of Drosophila looks like this. So it's a holometabolous insect, which means that it has 4 stages. So egg gives rise to larva, larva gives rise to pupa, pupa gives rise to adult, and from adults you get the next generation of eggs. So in terms of the dynamics, there are 3 major feedback loops which control the dynamics of Drosophila populations. So what I am showing you over here is about 70-80 years worth of understanding which has gone into creating this one little diagram that I am showing you. So if you look at the egg number, then that determines the number of larvae. Obviously, not all eggs hatch, but as far as we can see that there is no density dependence over here. Typically, a fixed fraction of eggs is what ends up hatching. Very late, I mean last 4 or 5 years, people have actually questioned this part, because they have actually ended up showing that there is egg cannibalism in Drosophila. Okay, so with that happening, then now one can actually put one more arrow over here, but the conditions under which the egg cannibalism happens is so limited that for all practical purposes you can disregard that arrow and think of this as a density independent process. So a fraction of the eggs will die and the rest of them will become larvae. Now once you have the larval stage, then 2 things happen. So in Drosophila, the larvae, it undergoes 3 instars. Instars are larval stages, and after that they become the pupa. Now there is a certain critical minimum mass that the larvae have to attain before they can pupate successfully. What does it mean? If the mass of the larvae at the time of pupation is less than that critical value, they will pupate, but they will not come out as adults. So they will die as pupae. Whereas if the mass is larger than that value, then they will pupate and they will come out as adults. So suppose you have a certain amount of food in which you are putting your larvae. Now as the number of larvae is increasing in the population, what is happening? The amount of food available, larval food available per capita, is going down. When that is happening, if it turns out that the larvae are not getting sufficient amount of food such in order to get to that critical minimum size, then they are going to die. Now if you are modeling it in a very simplistic way, you will simply say if the larval food is above a certain threshold, then everybody is able to attain critical minimum, because food divided by number of larvae, the per capita is above a certain threshold, and therefore everybody becomes an adult. Okay. However, in biology we know that that doesn't happen. In biology we know that there is variation among the larvae in terms of how much weight they gain, which in turn is determined by a bunch of other factors. We'll not necessarily take those into consideration over here, but essentially there is variation among the larvae in terms of how much food they gain. So what you end up getting is a distribution, and a distribution of larval body sizes, and all those larvae which are heavier than that critical minimum size, only they are the ones who will come out as adults, everybody else will die. Okay. Now, as you keep on increasing the number of larvae for a given amount of food, what will happen? The fraction of larvae which are on this other side of that critical minimum size, which means which are heavier than the critical minimum size, that will keep on coming down. And therefore, you have a larval density dependent larval survivorship. Okay. So this is this arrow over here. This is what it is saying. All right. Does this make sense? Everybody?
Yes, sir.
Yes, sir.
Okay. Okay. So now this larval number is therefore obviously going to determine what is the number of adults, but it is going to determine that through pupal survivorship. Okay. So that's what is meant over here. Now there is one more thing that happens. So the larvae, when they come out from the eggs, they're very small in size, tiny in size. Now as they eat, their body size becomes larger and larger. Now you know that there is a threshold, the critical minimum that I was talking about, below which they won't come out as adults. So that threshold essentially serves as the lower threshold of the adult body size. Okay. However, the larvae don't stop eating as soon as they reach that threshold. In reality, they continue eating well past that stage, and therefore you also get a variation in terms of the adult body size, which depends on how much food they ate as larvae. Okay. Now, the adult body size is an indicator of how much resource they have. So in Drosophila, typically, until and unless you give them a explicit protein supplement, typically the number of eggs that a Drosophila adult will lay will depend on what is its body size, which is dependent on how much it ate as a larvae. Now, when you increase the larval density, then the amount of food available for the larvae to eat after the critical body size is reached, that amount goes down drastically. And because that amount goes down drastically, the body size distribution of the larvae, that also shrinks. Okay, essentially you don't have too many adults which are, let's say, heavier and so on and so forth. You have most adults which are critical minimum, this is the lowest physiological possibly body size, and let's say a little bit ahead of that. When that happens, when their body size goes down, then the number of eggs that the larvae can lay, that the adults can lay, also goes down. So that is this loop: larval density dependent adult fecundity. Okay, so the higher the larval density, the lower the adult fecundity. And then you have a third loop operating, which is adult number dependent adult fecundity. So it is known that when you have Drosophila holed up in a vial or a bottle and so on, then beyond a point, and that point is actually pretty low, as you increase the density, the adult density, the per capita fecundity goes down. Why is that? That is supposed to be because of two reasons. A, remember the bottles, the vials in which you are keeping your adults, those are closed systems. So these guys are eating the food in the vials, but they are also excreting in it. So the amount of various kinds of waste products in the environment of the larva of the vial goes up drastically with increasing adult number, which is leads to a reduction in the adult fecundity. The other thing that happens is that as the numbers increase, there is physical interaction amongst these adults, and that also is supposed to reduce the amount of the per capita fecundity of these flies. Huh. Anybody? Anything?
Sir, is there a difference? Is there a difference between fecundity and fertility as terms?
Here it's basically how many eggs they can lay. Period.
Okay.
Okay.
Sir, as a
sir,
as a like a basic question, what sort of food do these Drosophila eat?
Okay, so in nature, typically they eat rotting fruits. Okay, here in labs, we give them different kinds of foods. It depends on which lab you are talking to.
No,
So
no.
for
I mean, can we control the parameters of food that we are using, like proteins or things like that?
yes. That's exactly what I am coming to. So you can see that by controlling the amount of food, remember this density that we are talking about is density per unit food, right? So automatically you can see that as soon as you control those densities, so many things can happen, right? The number of larvae will go down because lots of them will die. When that happens, then the adult number will go down. At the same time, their fecundity is going to go down. Okay, and if by chance the fecundity goes up because the density is less, then adult number, actually there is another feedback loop over here which brings down the adult fecundity. But then you can kind of play around with that by giving more food over here to the adults. All right, so you can see that remember I said that we are talking about life history, interaction of life history with nutritional availability, which basically means food availability. You can see how food is coming into the whole picture, right? So
does this also depend on the type of food that the larva eats, or only the amount?
So it actually depends on the type as well. However, for the sake of simplicity in these experiments, we give only one kind of food. But yes, you are absolutely right. Otherwise, if you do the same experiment on different kinds of food, you might end up getting slightly different results. Okay, to what extent they will differ will depend on how different the food qualities are. Okay. Anything else? Okay. So this is what our model looked like. So there are no equations in this one. This is just a schematic of the model. So each individual lays eggs. So you start the model with the run with some eggs, and all the eggs taken together is num egg. These eggs hatch, and this is density independent. So a fraction hatchability of them hatches to form larvae. So the total number of larvae is what is called num larva. Then you have food in that environment. So num larva and food will interact with each other to create density. Now, once you have this, then you essentially assign each larva a body size by pulling it from some kind of probability distribution. And then if the body size that has been assigned, some fraction of is going to be less than MC. So whoever gets a body size less than MC, they are assumed to be dead. Whoever gets a body size greater than or equal to MC, they are the ones who are supposed to be alive. So they form the adults. So when they form the adults, then you give them, assign them a sex simply by tossing a coin. Now you can play over here. If you have an unbiased coin, then you are giving equal probability of males and females. But if you want to study the effects of sex ratio, as we did in this particular study, then you can bias your coin and say 40% male probability versus 60% female probability, and whatever way you want to do it. And then you get your males, you get your females, and the total number is what is called num adult, and this num adult is leading to the adult density, and then these females, they are getting affected by the adult density, and they are also getting affected by what kind of food and to what extent are you giving to the adult, and then these two together is going to determine how many eggs each female is going to lay. So you count how many eggs each female is laying, and that leads to the num egg for the next generation. Okay, this is the schematic of the thing, which is very close to the biology, but that's what these kind of models are supposed to be like. So what are the various features of this? A, as you can see, it's stage structured, it's individual based, that means you are tracking each individual larva in an adult and so on and so forth. Actually, individual based models have different levels of detailing. So this is a model which is actually a fairly coarse model, it's not a fine model, and discrete generation, and it has important life history traits, measurable traits like probability of hatching, that critical mass that I talked about, the body size of the adults, how many eggs they are laying, and so on and so forth. Now, interestingly, these life history traits that we are using, these are actually very well-known life history traits that exist, that are common for most holometabolous insects. So we are not taking some very peculiar kind of a life history over here. So this is the same schematic, but now all the equations are incorporated over here. All right. So for example, remember I said that from number of larvae, okay, that is going to determine what kind of you guys have to ignore the phone, it's too far away and I can't reach it. So anyway, so this number of larvae, that gives that interacts with how much larval food is available to give the larval density, and from that you figure out the mean of the size distribution, and it is from this distribution that you pull your larval body sizes. So that one equation is over here. Then this is the equation which takes how much nutrition is available to the adult and maps it to the fecundity of the adults. Okay, and yeah, so that is that, and this is the one which takes care of the adult density dependent effect on adult fecundity, and you combine these two to get the number of eggs in the next generation. Again, the forms of these equations, these have been arrived at through extremely painstaking experimental work, and there are very good reasons for which these equations have been preferred over many other such possible forms. So anyway, now there are two major things over here. There are two densities. One density is larval density, the other density is adult density, and in both cases food is an important component because the density ultimately matters through food. So you can control the amount of food that you are giving to larva, and you can control the amount of food that you are giving to the adult in form of yeast. So for adult, if you give them live yeast paste, then their fecundity goes up. Okay, so you can do that at this stage. So for the sake of simplicity, you can take this part as, let's say giving yeast versus not giving yeast. So if you do all that, then you can create 4 different kinds of population dynamics regimes. What are these? You give a lot of food to larva versus limited food to larva, low food to larva. So you have 2 larval conditions, and you either give yeast or you don't give yeast. Okay, so when you have a lot of food to the larva and when you are giving yeast for the adults, you call that regime a HH. So high larval food and presence of yeast is high, so high adult nutrition, so high. Similarly, you give a lot of food to the larva but you don't give yeast to the adults, that's what you call HL. You don't give any food, or rather you give very little food to larva, but you give a lot of food to the adults, and you give low food to the larva and you don't give any food to the adults. Okay, so these are like 4 discrete corners. Now obviously you can also make them, these things quantitative, right? I have taken 4 corners as qualitative things, but you can also make the whole thing quantitative by changing the food amounts in small steps. We have also done that bit. So what we did was, we first did a massive experiment, a 49 generation long experiment. Now just to tell you what this represents, in Drosophila, the way these experiments were done, each generation is each year is 17 generations. Okay, so when I say 49 generations, this is 3 years worth of work, just 3 years worth of maintaining these populations. And for each one of these regimes, HH, HL, LH, and LL, we had 8 replicate populations. So we have 8 replicate time series for each regime. This is actually the second largest and second most detailed time series in the whole field, okay, of Drosophila dynamics. So this is the nature of our data, and from this we looked at 2 things. We looked at the distributions of the population sizes, and we looked at the fluctuation index, that same quantity that I talked about yesterday. Okay, then what we did was, remember we have 4 sets, right? 4 different regimes. We took 2 of those regimes, we took the HH and the LL, and we used those as training data set. So you know what is training data set? Okay, so when you have a model and you want to see whether that model is doing good for your dynamics or not, whatever it is that you're trying to predict, you run into a particular problem. What is the problem? The problem is that the model needs to be parameterized. Until and unless you parameterize it, you don't even expect the model to work for your data. But then if you parameterize a model for your own data, then very obviously that model is going to be a very good fit for your data, because you explicitly played with the parameters to make it so, right? Therefore, in data science, well, in population ecology, lot before the term data science became fashionable, what they do is they create 2 sets of data. So basically whatever data they have, they divide it into 2 parts based on whatever logic, and they use one part of it to parameterize the models. Okay, so the part of the data set that is used to parameterize your model, that is what is known as the training data set. And then once they have got the parameters, then they use those parameters to see whether the rest of the data set is fitting well to the model or not. So then the rest of the data set is known as the test data set. Okay, so what we did was, we took the 2 corners, the HH and the LL, and we used these as a training data set, which means we tried to make our parameters such that they fit these dynamics the best. And once they did that, then note, between HH and LL, you actually also cover the conditions for HL and LH, right? So we did that, and then we actually took these parameter values and tried to see whether that is doing well to match these dynamics or not. So we started with the distribution of the population sizes. Now, why did we start with that? We started with the distribution because in the previous paper we had shown that if you actually look at the distribution of the population size, then that is an outstanding how do I say, predictor of why the dynamics of the population is the way it is. Okay, so it has a very good prediction power for the dynamics, which is why we did that. Have you guys studied box plots? Do you know what box plots are?
Not really.
Not really? Okay, fine. Okay. So you guys know what a median is, right?
Yes, sir.
Yes, sir.
So you know what a mean is. A mean is simply the arithmetic average. Sum all the values and divide them by sample size. That's your mean. Then you also have what is known as the median, in which you take all the values, arrange them in ascending order, and whatever is the middle value, you call that the median. Okay. And if there are two middle values, then you take the average of those two. That gives you becomes your median. Now you also have what are known as percentiles. So what is a percentile? So a percentile is a value which divides the data set into two parts containing that fraction above and 1 minus that fraction below. Sorry, the other way around. So I'll just give you an example. So suppose we are talking about, let's say, the 25th percentile. Okay. So the 25th percentile is that point which divides the data in two parts such that 25% of the data is on one side and 75% of the data is on other side. Okay. Similarly, the 75th percentile is one that has 75% on one side and sorry, the other way, 75% on one side and 25% of the data on the other side. Okay. So that is the meaning of the concept of percentile. So by definition, the median is the 50th percentile, because median is dividing the data in such a way that 50% on one side and 50% on another side. Okay. So you see these things here. You see these stuffs over here, these bands. These bands are the medians. Okay. These red things that you see, these are the means. All right. This box that you are seeing, this box, the lower part of the box, so let's say this one, the lower part of the box is the 25th percentile. The upper part of the box is the 75th percentile. Okay. So between the 25th percentile and the 75th percentile, by definition, is going to be 50% of your data, the middle 50% of your data, right?
Yes, sir.
Yes.
So this part is what is known as the interquartile range. Okay. And then this one is the 10th percentile, and this thing over here is the 90th percentile. All right. So essentially, if you look at this box plot, I'm showing you a large number of percentiles: 10th, 25th, 50th, 75th, 90th, and so on. Okay. So these are all various measures, and together they tell you how the data is distributed. So for example, if you look at this one, okay, in this one, do you see that the mean and the median are very, very close to each other? Okay.
Yes.
And when the mean and the median are very close to each other, then both sides you have roughly a similar width. Okay. So this is a signature of a normally distributed data, a non-skewed. You know what is skew, right? A skew is, so a normally distributed data is one which will be like this. So it will be symmetric across the mean. And a skew is when you get something like this. Okay. When this is the median over here, this part is the median. So 50% of the data is bunched in a small zone, and the remaining 50% of the data is spread over a longer zone, and so on. Okay. So this thing that I'm showing you over here, this is equivalent to this one, whereas this stuff over here that I'm showing you, this one is equivalent to this one. Okay. So essentially, just by looking at the box plot, you can figure out what is the nature of the frequency distribution of population sizes, which, as I said, is very important because it allows you to say a lot of things about the dynamics. Okay, is this clear? So, bolo.
How many data points are there in each? Do these each of these vertical bars represent? Around.
So remember I told you that there were 49 generations.
Yeah.
And remember I told you there are 8 replicates in each regime. So 8 into 49, a little less than 500. No, a little less than 400, 392.
Okay.
Okay. So that's a hell lot of points to make a distribution. So anyway, so what you see in these white boxes, okay, these are the experimental data, and what you see in the gray boxes, this is the simulation data. All right. Now these two, we used them for training. So in some sense, we went out of our way to make sure that these two match. But then we took these parameter values and used them in toto over here. Therefore, look at these matches. As you can see, overall, these are very, very good matches between the simulation and the experiment. Okay, and the simulations were done exactly like the experiment and have exactly the same number of points in them. So you can see that the
Just one question. Why did we use LL and HH as a training data sets instead of splitting each one of the four into half and half and doing it that way?
Because our data sets are not very large, no. So what if we do LL and HH? What are we getting? We are getting for low larva, low adult, high larva, high adult. We can use that information to take care of this part.
Okay. Okay.
Okay. Now, if you use half and half of it, see, I'm not telling you about the training procedure. The training procedure itself was a very massive procedure, and it involves a lot of matching with things like autocorrelation coefficients and other stuffs. So if you take half and half, then the total time series that you end up getting for each is just about 25 or so. Okay, from those probes will not be constructed as efficiently as
Why do we need wait, are you saying each time series is halved, or you're saying the number of time series is halved?
Huh? Sorry?
Are you saying the
I assume that when you made half the data set, you meant that you take half of each, no?
Yeah. Yeah. So out of 8 replicates, I'm saying take 4,
Oh,
4, 4.
Oh, you meant take 4, 4, 4, 4. Okay, okay. No,
Yes.
that would have been a possibility, but then what will happen is, in some sense, we are including all the dynamics in our training. Whereas in this particular case, we are excluding two dynamics entirely from our training.
Yeah, yeah, okay.
So that is a much stronger, much stronger test. Okay. So then we also.
Excuse me. So what are those white dots up and down? Are those fifth percentiles?
No. Those white dots are outliers. So if there are any points which are beyond that level, beyond the 10th percentile, then you consider them as outliers. Okay.
Okay.
So that's what they mean. Okay. So you can see that even when you look at the fluctuation index, which is a measure of the stability, then this is the, if experimentally speaking, this is what you expect the trend to be: LH to be highest, and then HH to be second highest, LL to be third highest, fourth, HL to be fourth highest, and essentially you get exactly the same picture when you get it when you do the when you look at the simulation data. So we did a lot of other stuffs, okay, and essentially ended up showing that the model is capturing the dynamical features of the empirical data both qualitatively as well as quantitatively across all the regions. So now the question was that now that you have this model, what do you do with it? So what we did was we changed various parameter values. So basically we asked the question, what will happen if the life history changes? So remember, I was talking about getting mechanistic understanding about the system. So this is the mechanistic understanding part. So what we did was we saw what will happen if you alter the values of hatchability, alter the values of that threshold, alter those two sensitivities, to adult density, adult size, etc., etc. None of this is very critical, except that here we ended up getting some very nice, interactions between how the life is changing the life history, the pattern will of population dynamics will end up depending on which nutritional regime you are putting them in. Okay, so basically how much of larval food versus how much of adult food. So one very nice prediction that came out of this was that if you end up increasing the MC, the critical minimum of the larvae, then the fluctuation index is expected to go up monotonically, irrespective of which food regime you are talking about. Okay, now this was a nice prediction that could have been tested, and it so happened that right at that stage we had a population whose MC had drastically come down because of some other selection. Okay, so there was a completely different study, a different set of populations, a different set of PopDyne experiments, and in that we so these are the control populations. So these are the populations on which the previous PopDyne data was created, and from these populations another set of populations had been derived through selection in which the MC had come down. So what we did was we made these populations run population dynamics experiments, and we saw that in the LH regime, experimentally speaking, this is the fluctuation index of the controls, the JBs. This is the fluctuation index of the selected populations. This is the fluctuation index in the HL regime. This is the fluctuation index of the selected population. So remember, the prediction was that as MC reduces, fluctuation index will reduce. And then we actually took our model and simulated them for these corresponding things. And if you look at it, the correspondence between the experiments and the simulations are actually pretty high. Okay, I mean, in the LH regime it is superb. In the HL regime, the trend is captured, although the values are slightly off. So, and then we also looked at the population size distribution. And again, all the white ones are the experimental ones, all the gray ones are the simulation ones. And you see that by and large, we end up capturing their relative trends pretty well. So taken together, what we would like to say is that the model prediction seems to be matching very closely with the experimental observations. There were some other insights that don't really matter too much for the sake of this class. What I wanted to give you guys over here was some kind of an demonstration of how mechanistic models are constructed and how one can go about getting insights from them, which then can be used for further studies for asking other questions. Okay, so I'm skipping a lot of mechanistic details over here, but I hope that the illustration of the basic process is making sense to all of you. Okay, so I think we are very close to the end of the class. So I'll take just one or two questions and then I'll dissolve the class, but I can hang around for some more time if you guys want. You are getting the, what is it called? The video itself. So the video has all the information. If you guys want, I can share the paper with you, the paper which contains all the mathematical equations and where the equations came from, based on what experimental data and how the model was calibrated and so on and so forth. Oh my God, this is a very, very, very long piece of work. Okay. It's not the first model, obviously. We tried many things. So, okay, let me give you the backdrop to this model. This work starts with a model in 1988 by a guy called Larry Muller. Then based on that model, there were certain predictions. So that first experiment was launched. So that experiment was launched just before I started my PhD. And then during my PhD, I played around with this particular data a lot. And then I didn't make this new model that I shared with you guys, but I did other stuffs with the data. Then when I joined this place in 2007, 1 or 2 years after that, a IISER Kolkata student came here to work with me, and he happened to choose this data set and he happened to choose this model. So from 2008 to 2019 or 20, whatever, some 11 years, that student and I worked on this model for those 11 years. Okay, he finished his master's from IISER Kolkata, then he joined my lab as a PhD student, and throughout his tenure over here, he continued working with me on that. And then finally in 2019, I think we ended up finalizing the thing and publishing it. So yeah, it's a very, very long thing. Okay, I think I will stop the recording right now so that people who want to leave can leave, and then I'll answer all those questions.